Hello.
Ajay, good morning. Good morning to all. Good morning, sir.
Good morning, sir. Very good morning.
Very
So, good morning to each one of you. I, Dr. Vijay Kamal Meena, as a training coordinator, welcome each one of you to this training titled as Advanced Genetic and Plant Breeding Data Analysis Workshop. So, we are privileged to have our chief guest, Dr. D.K. Yadava, sir, the Honorable Vice Chancellor of Agriculture University, Jodhpur, Dr. B.R. Choudhary, sir, our officer in charge, Dr. R.R. Bharadwaj, sir. Your presence adds immense value to our gathering, and we are truly grateful for your support. Respected sirs, in this training, more than 250 participants have registered from various corners of this country. Participants are mainly faculties, research scholars, students, and scientists from various institutions, sir. So this workshop is not just an event; it's an opportunity for all of us to delve into the latest development in genetics and plant breeding data analysis. So over the next few days, we will explore a wide spectrum of topics ranging from basic statistical techniques to cutting-edge methods such as GWAS, genomics, and some bioinformatic tools also. So this training aims to equip all the participants with both theoretical understanding and practical skills that are essential in the ever-evolving landscape of genetic research. So, without any further ado, now I request Dr. R.L. Bharadwaj sir for welcome address to our chief guest and his remarks. Dr. R.L. Bharadwaj sir.
Thank you very much, Dr. Vijay Kamal. Good morning to all the participants. It is my pleasure to welcome all the participants to this online workshop. We are honored to delight to have a distinguished personality among us today, someone whose presence in add a special touch of grace in importance of this occasion. We extend our warm welcome, Dr. B.R. Choudhary, Honorable Vice Chancellor, Agriculture University, Jodhpur, and our esteemed chief guest, Dr. D.K. Yadav, ADG Seed ICR, New Delhi. Dr. B.R. Choudhary, having a vast experience on this subject, especially on nutri-cereals and superfood crops, and also as served as a vice chancellor in second times at Agriculture University, Jodhpur. Dr. Yadav, on behalf of ARSS Sumerpur, I extend our heartful gracious Dr. Yadav for a gracious presence in today with him. Dr. Yadav, not a name of, it is a synonyms of excellency and achievements. He has 25-year vast experience as Brassica breeder and including 19 year at ICAR New Delhi. The numbers of varieties, including Pusa Mustard 25, Pusa Mustard 27, Pusa Mustard 28, or so many other important varieties are released by hand of Dr. Yadav. At present times, he also engaged in different societal work as well as a breeder for improving the Brassica or Brassicae family, different species. At present time, also serving as a ADG, a seed post at ICR New Delhi, and also achieved so many recognized or prestigious award by Rafiq Ahmad Award and another Dr. B.P. Paul Memorial Award and NAS Recognition Award. So we are happy. We are really feel very lucky to at the presence of Dr. Yadav with him or our distinguished participants. I also convey my sincere thanks to Dr. Vijay Kamal Meena and Dr. Kapil Choudhary. Both are engaging in this event since long back, and really their distinguished work is very appreciable. Those are connecting to a community of the genetics or breeding and discussion on advanced genetics and plant breeding data analysis workshop. This is a really a very needful or essential workshop because the data analysis is a very important part of the research as present times it is compulsorily known all the participants. And again, I thanks to all the participants are enjoying the workshop from today to 3rd November 2013 and learn more and more about on this specific topic. Thank you very much, Dr. Vijay Kamal Meena and Dr. Kapil Choudhary.
Thank you, Dr. R.L. Bharadwaj, sir, for introducing our chief guest and vice chancellor, and for your this remarks. Now, I request our today's chief guest, Dr. D.K. Adava, sir, for his remarks. Sir, please.
Thank you, Vijay, and thanks to Dr. Bhardwaj for very nice introduction. Honorable Vice Chancellor of this esteemed university, Dr. P.R. Choudhary Sahab, Dr. Bhardwaj Sahab, and the organizers Vijay and Kapil, and the other faculty members, deans, directors, and most importantly, all the participants. So this today's the opening of this workshop on advanced genetics and plant breeding data analysis workshop. So, in fact, for at least those who are involved, whether it is a plant breeder or the agronomist who are involved in the conductance of the trials or handling of the large number of quantity of data. So, for them, this type of training, I think that is very essentially required, and many of our colleagues, the new scientists are joining. Sometimes these things are refreshed, and because there are large number of rapid actually progress, improvement in the various tools for the statistical analysis. So that's why the updation of the knowledge from time to time and understanding and knowing about the various tools for the statistical analysis, that is one of the very important component. And in that direction, the attempt made by the university and particularly the Sumerpur Agriculture Research Station of this esteemed university, that is very important. You all are well aware that earlier the data, when I think before us, myself and Dr. Choudhary, we are almost contemporary. So before that, people were doing most of the things were that data handling, data analysis that was being done manually, and the various designs. When I was actually posted, my first posting was in Sri Ganganagar, and when trial data was to be sent, so it was a Hercules task for minimum 7 8 days taking the trials. By earlier that facet machine was there. After that different the calculators they came later on. Earlier only simple calculator plus minus they were there, so most of the things they were being done manually based on the whatsoever the formulae and other things were there in the books, and it used to take a huge time. And the precision, if we talk, that was also not to that high level which we are receiving now. Later on, the scientific calculator came, so things were little bit eased down. And after that, the in computer, when MS Office came, so Excel was one software where we generally used to work for the various trials, data analysis. But the very important data analysis like the various breeding trials in Delhi, in line tester and others. So they were generally very difficult, and people used to depend on a few people in each university, and the ISRI center was there. So a long queue used to be there for getting the data analyzed. But with the development of the technology now, and with a very I think rapid progress in terms of the statistical analysis tools, and ISRI being the nodal institute and having a large number of trainings, development of a huge number of the softwares which are easy to handle and which can be done, which can be handled once after a very short training, and the availability of the various PCs. Everybody is now having the computer on their table. When in 1998, I remember myself and Dr. Choudhary, we went for a Irish training at NIC in Delhi, and we used to stay in IRI Farmers Guest House. So that was our first exposure to the computer, in fact, at NIC. And at that time, there was the coordinator was one brigadier was there, Brigadier Sharma. So in 1998, he used to tell the time will come that your phone number, mobile was never talked at that time, that your phone number will remain same where so you go, so you will be able to attend the calls and other things. So that was a actually very surprising thing, and we used to generally think that how these things will happen. But you see the development in the information technology, in the statistical tool, and now with the advancement of the various tools which we are using now, the marker assisted selection we are using, the Jivas as Vijay was telling, And bioinformatics has actually become a very strong tool, and a large number of, I think, programs are there. So for that, the awareness of such through such type of programs is essentially required. And the uh, I think the number of participants Vijay was telling 253. So that is a good, I think, response from all the scientists. And a number of the models and designs are also being used in the crop improvement science, not only in crop improvement, but in other disciplines like NRM, engineering, animal science. So everybody is using the new tools and techniques, and for that, particularly for the its statistical analysis, to come to the conclusion, to have the good inferences out of that data. So this type of training is essentially required, whether you are getting it through the ISRI or some of the professional person, but at least this training will help you to understand the data, and it will always be helpful in drawing the inferences, drawing the conclusions out of the data which you have done, and the precision which we want for the results, and further output and outcome which will come out of that data which we have analyzed, and in publication of good peer reviewed journal papers in the journals. So for that, definitely this type of training is essentially required. And I am sure that the faculty whichever they have selected, very competent faculty will be there for these 5 days, and they will be delivering the lecture. So to all the participants whosoever have joined, till now 139 participants are there. So my request will be that, not for the sake of having a certificate that I have attended this workshop for your academic purpose, whether they are scientists or even the students also. So kindly be serious and attend this 5 days training in full time, and have your queries during the various lectures when our faculty is delivering, so that you can have at least the better understanding, and in the times to come you can handle your data. And for your research paper writing or your report writing, you need not to depend on anybody, that I will go to somebody, he will be telling that how to write a report, how to analyze data, how to have the inference out of that. So I think for that purpose, it is very essentially required. It will make you totally self-reliant and enhance your knowledge. And for further advanced courses, if you have the basic understanding of the subject, so definitely that will be helping you in the future course of action also, when you go for some advanced training. Up to until you don't have the basic knowledge of any subject, so then going for some advanced training or handling your own data, so you always become dependent on others. So in that aspect, I think this training will be very useful for all the participants. I am sure the faculty, very learned faculty, they will be taking full time, and whatsoever the practicals are there, so they should also be emphasized more, so that people can understand, because learning by doing is more, I think, convenient and more effective. So in this direction, definitely the arrangements could have been made. And whosoever is attending, my only request to all of you is that kindly be full-time attendee, not that by opening the screen and you just put your camera off and you are working on some other aspects. So whosoever has registered, kindly spend the time. This is only for you, for enhancing your knowledge, making you actually knowledgeable in terms of the data analysis and other things. So, with these words, I think I thank the organizers, particularly Honorable Vice Chancellor Dr. Choudhary Sahab, for giving me this opportunity to interact with all of you. And I'm sure that a very good faculty will be there, and this training will be very, very useful to all those who have registered and will be participating. And once again, for a very successful training to program, and for very useful training to all of you. So, thank you very much. Thank you, sir.
Uh, Thank you so much, Dr. D. Yadav, sir, for your kind words and making this event more energetic with your presence. Now I request Dr. B.R. Choudhary, sir, for his remarks on training.
Thank you, Dr. Meena. Good morning to all. Dr. D.K. Yadavji, my friends, ADG SEED ICR New Delhi, and the chief guest of this inaugural function of 5 days training on advanced of genetics and plant breeding data analysis. Dr. R.L. Bhardwaj, Dean College of Agriculture, Sumerpur. My faculty, Dr. Vijay Kamal Meena, and Dr. Kapil Choudhary, who is the planner and designer and organizer of this training program. Dear participants, I welcome to all you on behalf of Agriculture University Jodhpur. Genetics and plant breeding is the main and backbone of agriculture sciences and has significantly contributed in enhancing the yield of different crops in the country. In the era of climate change and limited and depleted natural resources, its importance will increase for sustainable productivity of different crops. As per the need of experimental design, there are numerous experimental design and different process of data analysis for interpretation and results for the conclusions. I hope this workshop will help to participant in the planning of their experiment and data analysis using different statistical methods, tools, and software. Once again, I thank to all you, particularly Dr. D.K. Yadav. I thanks and express my sincere thanks for your presence in this function. Thank you.
Thank you, VC sir, for your word of wisdom and making this event more fruitful with your presence, sir. Now I call Dr. Kapil Choudhary for vote of thanks and introduce today's resource person for the training. Unmute, please unmute yourself.
Okay. Thank you, Vijay, and good morning, everyone. It is my privilege to extend a warm and sincere thank to each and every who has contributed to success of the inaugural session of our workshop. And first and foremost, our heartful thanks to Dr. D.K. Yadav, sir, ADG, for his gracious and invaluable presence. Sir, we are truly honored to have you in today's function. And I would like to special thanks to our Honorable Vice Chancellor, sir, Dean, sir, and I also extend my appreciation of organizing committee to ensure the smooth execution of this event. So thank you once again to each and everyone, and all the best to the participant. So thank you, sir, to join today's inaugural session. Now we will start today's session. And thank you, sir.
Okay, sir. Thank you. Thanks to
Thank
all
you so
and all
much,
the
sir.
best.
Thank you so much, sir.
ठीक है
So-
विजय.
Yeah, sir. Thank
Okay,
you so much, sir.
So today's first lecture is Introduction to Genetics and Plant Breeding, Use of Data Analysis, and it will be delivered by Dr. Subhash Chandra, scientist at IGFRI, Jhansi. So, sir, please start the session. And I request all the participants after the completing the session, we will upload all the lectures on a YouTube channel. We will provide your link so that you can access whatever the information we will provide you later at your convenient time. And after the completion of the session, we will provide a manual which consists of theoretical and practical session.
Dr. Subhash will join in 5 minutes. He will start his presentation. So
हेलो मैम लैब में हूँ। मैंने जब भी किया था सुबह अभी आया।
Sir, please unmute yourself. Subhash sir.
Sir, Subhash sir is coming in 5 minutes, sir.
Dr. Kapil, you continue. You continue. Just introductory remarks is also okay. Dr. Kapil. Oh,
Kapil,
Dr. Vijay
unmute.
Kamal. Dr. Vijay Kamal. Vijay Kamal, you continue. Continue the program.
Yes, sir. Sir, actually, he has joined, but he will start his presentation in 2 to 3 minutes only, sir. So
Okay.
we are waiting for him.
Okay. Okay. No, no, man.
Okay. Kapil, unmute, please.
Hello, participants. Okay, so जब तक सर आते तब तक we basic discuss about the today's class, what we will discuss in today's class, what will be outcome of the today's class. So the first half an hour or maybe 40 minutes we will discuss about the theoretical part of the data analysis in the GPP, and after that we will go for the practical session in which today only we discuss about the basic of the software. Tomorrow onwards we deeply go for the experiment or practical session which are used in your GPP. Maybe in thesis or somewhere else, maybe in research also. So under that, you may be definitely explore about the multivariate analysis, design of experiment, GWAS, bioinformatics, and all this. So, but to start today's class, we will, I think we will come across the what will be R is there, how to install R. I mail you one file is there in how to install the R. Okay, so before to practice over commands and code, you have to install the studio in your system. Maybe like if someone of you are using Windows, someone are using Mac and all this. So according to you have to install, and so that it will be easy to hands-on, like it will become a hands-on training for each and every participant. And basically why we go for R, because we definitely will come across many software, but we mainly in today's session we will discuss about R. Why we go for R only? Actually, mainly R, what the name meaning of R? Mainly R given by Robert and Ross. They are two scientists at University of Auckland in New Zealand. So they from their name, initiation name, they have taken as a R software. R nothing but is a programming language is there. We also work in Excel. The same way we have to work in the R programming language. And it mainly derives from different language, like it mainly derives from the S programming language. So it is also initiation of their scientist name and also taken from S programming language. So why we go for only R? There are two benefits of the R. First, it is open software. Open software in the sense anybody can install it and use it, first of all. Second, for its better graphical representation. Whenever we go for the analysis on everything, because a picture says more than the thousand of words whenever we read some type of quotation. So that's why for the two benefit, we go for that. Second benefit is suppose I develop a package, okay? Maybe I'm expecting time series modeling and forecasting. So if I develop a package or I give a modeling, I will upload my package in the R. Crane is there, comprehensive R archive network is there. So I will upload my packages there. So there are existing now nearly 20,000 of packages are there, installed packages are there. They are most of them are installed by different person of their expertise person are there. So are mainly examine all these packages to upload in their site. Before to upload their site, they mainly examine all the packages thoroughly, and after that they will be upload their packages. So suppose I uploaded 10 packages. So in the area of time series focus, someone working in design of experiment, they will upload their packages also. So it is open software. So every expertise of that particular subject will definitely give an input to that, and it will directly benefit. Means there are 20,000 packages are there. Means we can go more than, I think, 1 million yearly analysis we can do in the R software. There are 20,000 packages are there, but in each and every package there are maybe 10 or 20 function are there, different function are there. So we will use different function and which are easy or which make smooth or analysis for a final presentation. So in statistics, we mainly do 3 or 4 work in statistics. We mainly call as a COPI. In COPI means first we have to collect the data, means collect data. Suppose I am going to for a design of experiment. So first of all, we have to reach the experiment. So first of all, we have to collect the data. Like I suppose I want to run a RBD design in R. Is there, sir? Hello.
हेलो
सुभाष सर,
हाँ
स्टार्ट कर रहे हो?
स्टार्ट तुमने स्टार्ट किया है क्या? हैव यू
नहीं
स्टार्टेड?
नहीं सर, मैं तो ऐसे जस्ट इंट्रोडक्शन कर रहा था. कोई नहीं, आप स्टार्ट कीजिए सर एक बार, ठीक है ना? मैं आप प्रैक्टिस
मेरा स्क्रीन जो है वो विजिबल है क्या?
हाँ विजिबल है सर, आप आराम से स्टार्ट कीजिए सर. हाँ
Is it
विजिबल है
visible?
सर, हाँ विजिबल है सर. सर आप वीडियो ऑन कीजिए अपना एक बार, एक. हाँ ठीक है सर, हाँ ठीक है सर. ओके. So now Dr. Subhash, scientist IGFRI Jhansi, will start today's lecture on introduction to genetics and plant breeding data analysis. So sir, floor is yours, sir.
Thank you, Dr. Kapil and Dr. Vijay, for inviting me in your training program entitled as Advanced Genetics and Plant Breeding Data Analysis Workshop. So thank you for giving me the topic Introduction to Genetics and Plant Breeding and its importance through data analysis. Okay, now okay. The first thing we should know that as we are discussing about the genetics and plant breeding. So first we understand the genetics. Genetics, basically we study two things. One is the genes, second is heredity, and third one is the variation. So genes are nothing but these are the segments of DNA which transmitted from one generation to the next generation and carry the genetic information from parents to the progeny. So second important thing is that these traits are passed from their parents to the offspring. If there is any change in the DNA sequence, then we can see the change in the phenotype. However, the phenotypic expression can be influenced by genetic makeup as well as the environmental or climatic factors. So another important thing is the variation. How does dissimilarity between the children's or offspring, between the plants to plants, between the progenies, and their differences from their parents. So we must understand the variation. Why this variation occurs in the plants, in living organisms. So these are the few basic things which decides the variation, which creates the variation, like chromosomal rearrangement, mutation. We know the long-term impact of mutation. It might be the friendship, it might be the point mutation that influence the whole ecosystem of the living organism. And another important point is the crossing over. We know that crossing over is one of the main factor that creates variation among the offsprings. A little bit, it is about the historical part of the genetics. We know that Gregor Johann Mendel, and we know that he was born during 1822 and died in a short span, and he died in 1884. So basically, he's known as father of modern genetics, and we know his contribution in the genetics. First is the principle of dominance and principle of segregation. Both these principles usually work in single gene. So principle of dominance denotes the genetic function, whether one allele is dominant on another allele for a single trait or not. Or principles of segregation, generally it happens during the segregation, like during the recombination. So it leads to genetic transmission from parents to the offspring. And third one is the principles of independent assortment. We know that this principle usually works when we study two traits simultaneously and there is no linkage. So it is also denotes the genetic transmission from parents to the offspring. Before going to the phenotypic and genotypic data, we must know what is the genotype and what is the phenotype. So if you see the phenotype, whatever we see, first we are talking about the phenotype. If you see the physical appearance of an organism, we call it the phenotype, like plant height is a phenotype, like number of tellers is a phenotype. What we can see visually, or it is the characteristic of an organism, like you can see in the figure, purple color is encoded by homozygous dominant allele. In heterozygous, it is same like purple. But in case of recessive homozygous condition, it is showing up white color. So here clearly you can see that we don't know the genotype, but based on the phenotype, we categorize these plants into the different categories. If you see the genotype, it is a set of genes in DNA that decide the unique characteristics of an individual. It might be a plant, it might be an organism. So there are different branches in genetics. Normally, Mendelian genetics we call as classical genetics. So here you can see transmission of traits from parents to offspring. What Mendel did in his lifespan, we consider it as a classical genetics, which is mainly based on the phenotypic data. We don't exactly, he don't know the genotypic, what is which trait is governed by which gene, but based on the phenotype, he gave different concepts. Second one is the molecular genetics, where we study the chemical and physical properties of DNA, RNA, and protein. In case of population genetics, generally we talk about the larger size of populations, like in, you know, that we study the Hardy-Weinberg law. This is the population genetics. Mostly population genetics is covered when we study the large size of population. We don't talk about a small set of individuals in that. In case of quantitative genetics, we study the relationship between the genes and their encoded traits. In case of quantitative genetics, different types of we relate the genes with the phenotype. So another important thing is that it is the plant breeding. So we all know that it is an art because there is a need of human skill from sowing to the harvesting. We need human. I can say that human skill is needed from sowing to the harvesting. When we talk about the science, actually we need different things like which design we should use, what type of data we should take. So this is the science from planning to the use of proper scientific design to study a particular objective. And when science and art both combines, then there is a development of resources. We call it as technology. So three principles are required for effective plant breeding program. So this definition is plant breeding is the purposeful manipulation. Important thing is that we are doing manipulation in the plant purposefully. What we need, we require that one. Not all the traits are useful for human being. So we design our experiments what we want from them, and manipulation of that purposeful trait to create a new variety which have specific desired characteristics which are required for us. It might not be useful for plant, but for human being it is required. So there are the different objectives while recording the data. So some are increased crop yield. Crop might be, in case of fodder, if you talk, fodder yield is important. In case of sugarcane, if you talk, cane yield is important. In case of cotton, if you talk, ball yield is important. So these are the different. In case of wheat, grain yield is important. So increased crop yield is the ultimate target of any plant breeding program. We can't compromise with the crop yield. In each and every phase, we want to increase the yield. However, it is not possible to increase incremental increase in the crop yield, but it is the basic and fundamental objective of plant breeding to increase crop yield. Crop quality, it might be the crop quality. For example, in forage crops, crude protein is important. In case of, if you talk about the cane quality is important. So there are different quality parameters in different crops. So we want to increase these crop quality parameters without hampering the crop yield. Another important aspect is either you develop the resistance. If resistance is not possible, then go for tolerance for different biotic and abiotic stresses based on the requirement of crop, based on the severity of the biotic and abiotic stresses in a specific crop. So another important aspect is development of climate resilience cultivars, because you know that different challenges have occurred in the modern era. So we need to solve modern approaches to solve the modern problems. So it is needed to develop agroclimatic conditions based cropping system. So we need varieties like short duration varieties. We need to fit in the cropping system. We need different parameters. For example, maturity should be synchronous, one-time maturity. Non-settling type, it is needed in case of pulses. Photoinsensitivity, where crops are very much sensitive like wheat. So can we develop photoinsensitive lines, determinate growth in case of, again, in case of moong or like dals. So can we develop in case of cotton also, can we develop some determinate growth? So these are the basic objectives of plant breeding. So depends on the crop, we decide the breeding objectives. Now important thing is that what is the need of statistics? First thing is that what is the data? Data is nothing but based on your requirement, based on your objective, You decide the parameters and you take these data from your field. So these are the individual piece of factual information recorded. So this is the primary data. Secondary data, generally we use primary data for generation of secondary data. So in statistics, what we do, we take data, we arrange the data accordingly as per the need of the software, and we do the data analysis. And in result, you will get statistical analysis of different figures. So we do interpretation and presentation for that. So in case of statistics, it is the study of data and can be used to describe, predict, infer from a data set. So basically, it makes complicated data into easily approachable and easy to understand. So here there are the different types of data. You can see the qualitative data. Qualitative data are, you cannot categorize, you cannot give them like numerical values. You can't give them for qualitative data. For example, if you see that color, taste and gender. In case of gender, male or female, there is no other category. However, it is transgender is also, so you can categorize in 3 categories only: male, female, and transgender, like in taste. So these are the categorical data. So here it is divided into 2 forms. One is the nominal and another is the ordinal. So if you see the nominal data, it cannot be ordered or measured. What does it mean? Its mean is that if you say that blue is my favorite color, so this is the fix. It is the blue only. You can't say it is the red. So you have only one choice. Like, which is your favorite teacher? It will be only one. So these cannot be ordered or measured. Like, what is your favorite book? What is your favorite eye color? So this is the nominal data which cannot be ordered or measured. But in case of ordinal data, it can be it can have a natural order, but not necessarily measured. Like if you do the disease rating 0 to 9 scale, so you categorize your disease screening into the different scales. Like 0, it will be the immune type, or 1, 3, 5, 7, and 9. And based on your visual observation, you categorize them on a scale. Like rating, if you give the rating and ranking, which one is first, which one is second. So here you have a natural order, but not necessarily measured. It is not, it doesn't guarantee you that you can measure it. So this is the qualitative data. In case of quantitative data, we call it as a numerical data. We call it as a numerical data. So here you can see the plant height. It may range from, like in case of oat, it may range from 100 to 200. So variation is very high in case of weight, length, size. So in this category, you have 2 data. One is the discrete and second one is the continuous data. In case of discrete data, you have a finite value. A fixed value will be there. But in case of continuous data, there will be a continuous continuation in the data. Like if you see the temperature range, it might go from 30 to 40 degree. Like plant height, it is a continuous data. But if you talk about the number of students in a class, it might be 5, 6, it cannot be 5.5, 5.6. So this is the discrete data. But basically, in case of morphological data we study in the plant breeding, these are the continuous data. Like weight, it might ranges wide. So if you talk about the primary data, as I already mentioned, that it is collected by you in the field itself. But in case of secondary data, you use primary data to predict something from the primary data, and it is mostly used in surveys and research studies. When we talk about the univariate or multivariate data, so univariate have only one variable and multivariate have different variables together. So if you see the statistics, so we can use both univariate as well as multivariate data. So what are the challenges in data analysis? So first is to how to model genetic versus non-genetic variation in a large field trials. So it is quite difficult to generate a model for genetic and non-genetic variation. So another is the detect the QTL for complex traits, complex especially the quality traits. So like biotic, sorry, not for biotic, it is for biotic traits. So detecting the QTL for complex traits, it is very difficult. And sometimes it is what we predict, it might not be usable also. And another important, how to use genome-wide molecular markers to predict the phenotype of unphenotyped individuals which have not been phenotyped for that particular trait. How to model complex inheritance pattern in polyploids, because if you have diploid, then you have only two sets of genome, but in polyploids you have multiple, like in tetraploid, hexaploid, octaploid. So it is difficult to understand the inheritance pattern, and sometimes to correlate with plant physiology and peak phenotype data with the genetics. So there are different challenges in data analysis, or sometimes you have a big data, but you cannot analyze, or you cannot use the proper statistical design to generate more useful interpretation. So there are few importance of data analysis in plant breeding, like big data are generated such as phenotypic data might be sequence data, and pedigree data might be there. So it offers to accelerate the breeding process. Actually, based on your statistics, you can target your trait of interest. Like if you are screening the genotypes for biotic stress, then by using the statistical tool level of significance, then you can say that this genotype is more tolerance to, like for example, in case of mustard, particular X genotype is more resistance or more tolerance to virus disease. So based on the statistical approaches, you can underline that genotype, and you can understand the underlying biological mechanism also. And another important is validity and reproducibility. It is only possible through the statistical softwares. And one important thing is that when you are screening a large number of genotypes, so you can develop a small core which have a wide variation, and it is suitable for further studies. So from the large genotype, you can go to the small set of genotypes by developing the cores. And you know that different types of graphs and tables you get after the data analysis, so it represents in a better way. It provides you a better visualization and understandability of your raw data. So, for example, I have mentioned few phenotypic traits. Generally, we use in fodder crops like green fodder yield, dry matter yield, crude protein yield. First two are the based on the morphological data, and last five are, these are the quality parameters we use in fodder crop. I have also mentioned a few examples of wheat where you can see the spike length, number of spikes per meter square, grain mass per spike. So these are the phenotypic traits you take data in the field. So for this, you need different types of statistical design. So all of you know that basic principles in each experimental design is replication, how much replications you are using, whether you are randomizing them or not, and you are using the local control or not. So based on the requirement of the breeder, you either use RBD, or in general we use in plant breeding RBD and augmented design. If number of genotypes are more, then you go for augmented design. If number of genotypes are less, then you go for randomized complete block design. So mostly in the breeding we use them, but there are different statistical design based on the need of the researcher. They use them. So for example, I have mentioned one of design in case we use in oat, for the oat. So if you see here that this is the technical program we prepare while sending the material to the different locations. So here you can see that 8 number of genotypes with national and zonal checks. Zonal checks is also mentioned in that. We use design RPT with 3 replications, plot size, rows, how many rows we have mentioned. We mentioned in that technical program what should be the seed rate, fertilizer, and different seed requirement of different checks as well as entry. And these are the 27 I have mentioned here. These are the testing locations where we send the material. So basically, this is the formulation of design, how we prepare in AICRP, how we prepare the different designs. I have mentioned here only for ocean. So I have taken one example, data collection. For example, in IGFRI, we have studied 105 genotypes. However, I have mentioned only 4 genotypes here. Likewise, you can go up to 105 genotypes. So these are the 5 traits: germination percentage, root length, root shoot length, seedling biomass, and seedling vigor index. So it will go up to 105 genotypes. So when you put these data, you take the mean, and by using the software R, we have used we have prepared a principal component analysis. So basically here we use only mean, although we have done the design in RBD, but like replication 1, 2, or 3, we take the mean, and by taking the mean, using the your software R, we have prepared a principal component analysis. So you can see that different types of genotypes, not 5 genotypes and 5 traits. Simply this figure is presenting your data into a simple form. Like, for example, root length and shoot length, they are they are showing the correlation. So based on the Y plot, you can predict that which genotype is better. What are those recorded traits which are showing more correlation between them, and what are the genotypes which are having the similar grouping. So based on that, you can interpret your data. For example, the same set here, 15 traits are here. We have studied in case of sorghum, 15 traits and 12 genotypes. So we calculated the correlation. So here you can easily see that by using the software R, we have developed that. So this figure, the star, which is highlighted in red color, it is showing the correlation between the different traits among the 15 traits and 12 genotypes. We studied the genotypes in sorghum. So it is clearly indicating that the raw data where you cannot say that which trait is highly correlated to another trait. So based on the statistical tool, you can easily interpret that what are those traits, whether these are highly correlated with another trait or not. So a correlation might be positive or negative. Based on that, you can decide your breeding program in which direction you will go. Likewise, we have studied Cenchrus liris. It is a fodder grass. So, as I mentioned that we have different locations. So across the country, 22 locations and 9 genotypes. As you know that this is a perennial grass. So here I have mentioned only the crude protein content. So no, not that one. It is the sorghum. Sorry, it is mentioned. It is not. This is the sorghum genotype. Same 12 genotypes are there. So based on this data, we have developed the dendrogram. So these are the I think those charts. These are the different genotypes based on 15 traits. They were categorized into the different clusters. So through the R software, so here you can see that dotted lines are clearly indicating that genotypes grouped in that dotted line particular, they have more similarity than the other. So this is the clustering or dendrogram of the 12 genotypes based on 15 traits. In the another trials, this is the in the next trial, we have done the stability analysis, where I have mentioned that Cenchrus liris, it is a perennial trial. Trial was conducted at 22 locations for all 3 years. And here I have mentioned only crude protein. However, we took 5 parameters. So this is the GGE analysis. So we have, if you put the data into the software, then you will get a different types of graph. And for study the different, to understand the different types of data generated by our software, you must read different papers published for GZ biplot analysis. So here you can see the first graph is mean and stability. Here you can see that we have 9 genotypes and 22 enrollments. So data has been divided into 2 parts, PC1 and PC2, like the biplot analysis. So this graph will show you that which genotype has the higher mean and higher stability. In the second graph, you can see that it is showing you that biplot structure, same PC1 and PC2. So here it is showing that which enrollment is better, which genotype is better, in which location. And another analysis, another figure is discriminativeness and representativeness. If you do the GG biplot analysis, then you will face all these results. And for interpretation, you must read few review articles which have been published by the different researchers. So this is the ranking enrollments. Ranking enrollment is showing that which enrollment is more discriminated, which enrollment is more suitable for discriminating the enrollments over the year. Like ranking of genotypes, it will show you that which genotype is performing better and which enrollment. So this is the little bit overview. So thank you, Dr. Vijay and Dr. Kapil.
Thank you, Subhash sir, for excellent presentation. You have discussed very well about different types of data and how to interpret them, and some of the basic concept of genetics and plant breeding. And if anyone have any doubts, you can ask him. Any participants? So if there is no doubt, then we will go
Hello,
further.
sir.
Okay, yeah.
Namaste, sir. This is Yashpal Singh. Firstly, thanks for this wonderful presentation. Sir, I have one question. Sir, what is the difference between MEY plot and GGEY plot, sir?
ME and GG,
Yes, sir.
देखो ME के अंदर और GG के अंदर केवल जो एनरोलमेंट का इफेक्ट है, ओनली एनरोलमेंट का जो इफेक्ट है, जीजी के अंदर तो आप क्या करते हो, जीनोटाइप और एनरोलमेंट का इंटरेक्शन है
Okay, sir.
जीजी के अंदर। जब आप कोई भी एनालिसिस जो है वो आप दो पार्ट्स में कर सकते हो, या तो आप दोनों सॉफ्टवेयर का यूज कर सकते हो, ठीक है,
Okay, sir.
एमई का भी कर सकते हो और जीजी का भी कर सकते हो, लेकिन
Yes, sir.
आपको सबसे पहले ये देखना है कि आपने नंबर ऑफ ट्रेड कितने लिए हैं पहले तो। मान
Okay,
के
sir.
चलो आपके पास नंबर ऑफ ट्रेड 4 है,
Yes,
ठीक
sir.
है। अगर आप एमई से करोगे तो एमई के दोनों सॉफ्टवेयर लग जाएंगे वैसे। जब आप एमई भी
Okay, sir.
निकाल सकते हो, एक ही पेपर को, एक ही डाटा को आप एमई से भी कर सकते हो और जीजी से भी कर सकते हो। लेकिन अगर आप जीजी का यूज करोगे ना, बेसिकली डेफिनेशन के हिसाब से तो इनमें डिफरेंस है, शायद एनरोलमेंट का है मेरे हिसाब से। एक में एनरोलमेंटल वेरिएशन का शायद एमई में है और जीजी में नहीं है, लेकिन एक ही डाटा को आप एमई और जीजी दोनों से लगा सकते हो। अगर उसमें सिग्निफिकेंट वेरिएशन है ना,
Yes, sir.
तो दोनों ही लग जाएंगे। लेकिन बेटर ये रहता है कि अगर आपके पास नंबर ऑफ ट्रेड कम है, जैसे 4 या 5 आपने लिए हैं, क्योंकि जीजी से
Yes,
जब
sir.
आप एनालिसिस करोगे तो एक ही ट्रेड का
Yes,
आई
sir.
थिंक करीब 6 to 7 फिगर्स बनेगा। तो अगर आपने 4 ट्रेड लिए, अगर आप पेपर के अंदर उसको इनकॉर्पोरेट करोगे तो लगभग 28-29 फिगर्स बनेगा आपका, जो कोई भी जर्नल आपको लेगा नहीं। तो
Yes, sir.
बेटर ये रहता है कि अगर आपने रिसर्च पेपर इसके देखे होंगे तो फॉर एग्जांपल आपने 15 ट्रेड्स लिए, 15 ट्रेड्स हैं आपके, 3 आपकी मान के चलो, 2 या 3, नॉर्मली जब हम स्टडी करते हैं तो आपका या तो 1 साल का डाटा होगा या 2 साल का डाटा होगा। लोकेशन की भी बात
Yes, sir.
करोगे आप, लोकेशन आप ज्यादा से ज्यादा 1 करोगे या 3 कर दोगे,
Yes, sir.
या तो आप एक लोकेशन पे 2 साल का डाटा हो गया, या का 1 ईयर है लेकिन 2 लोकेशन का डाटा हो गया। तो बेटर
Yes,
पे
sir.
जो है ना वो है कि आप जीजी से ही जाओ,
Okay, sir.
क्योंकि जीजी से इंटरप्रिटेशन बड़ा अच्छा आता है आपका। जैसे विच वन वेयर है, कि विच जीनोटाइप इज सुटेबल इन विच एनवायरमेंट, वो आपका एक अच्छा
Okay.
इंटरप्रिटेशन देता है उससे कि आप किस जीनोटाइप को लेके फर्दर ब्रीडिंग प्रोग्राम में यूज कर रहे हो। तो मेरे हिसाब से तो जीजी जो है वो ज्यादा लोग यूज करते हैं बजाय एमई के।
यस सर।
सर, एमई के अंदर भी अपने ये पता कर सकते हैं कि कौन सा जीनोटाइप किस एनवायरनमेंट में बेटर है, जहाँ
नहीं
उसमें
है,
ये
ये नहीं है उसमें,
ये
एमई में
लिमिटेशंस
नहीं
है
है।
एमई की। जीटीवाई प्लॉट के अंदर जैसे बिच बोन वेयर प्लॉट और भी दूसरे सर जो ग्राफ्स वगैरह जितने बता रहे हैं ना, मतलब जीजी प्लॉट के अंदर आपको स्पेसिफिक मतलब किस एनवायरनमेंट के अंदर है, कौन सा जीनोटाइप है, कौन सा एनवायरनमेंट ज्यादा स्टेबल है, तो ये सारी चीजें फैसिलिटेट करता है जीजी बाई प्लॉट। एमई की थोड़ी
ओके
रेस्ट्रिक्शंस
Shall I
होती हैं
add?
उस ये
Yeah.
ओके सर, थैंक यू
Actually,
सर.
both are. Hello. Shall I add?
हाँ जी बताइए।
Yeah. Please. So both are stability analysis tools, and both are said to be complementary to each other. Means depend करता है कि आप किस टाइप की analysis कर रहे हो. And ME में आपका environment effect होता है. Moreover, it is a combination of principal component analysis and ANOVA.
जी
यस
So
यस मैम.
somewhere it is found कि ME is better than GG, and somewhere it is found that GG is better than ME. So if you'll go to the research papers now, you'll find that both are complementary to each other. So if you're really planning for your PhD thesis or something like that, so I will suggest if you can analyze both GG and ME in your data, then go for both. Then that way you will be able to give more good analysis, plus 2 analysis you are giving at the same time. So that would be more beneficial for your research program as well. So
Okay.
there is no proper comparison between ME and GG both. Both are complementary to each other as far as I have studied. Yeah.
लेकिन एक
Okay,
चीज
okay.
है,
Thank you.
ज्यादातर या तो आप, मैं तो जितना रिसर्च पेपर देखे हैं, या तो लोग एमई की तरफ जाते हैं या जीजी की
Yes.
तरफ जाते हैं। दोनों एक साथ कोई भी रिसर्च पेपर कम ही मिलेंगे आपको जिसमें एमई और जीजी
हाँ
दोनों
सर,
का उन्होंने यूज करके और अच्छा पेपर पब्लिश किया।
हाँ सर, एक्चुअली ये सिर्फ कंपैरिजन मींस आई एम जस्ट टॉकिंग अबाउट द कंपैरिजन दैट
जी
यू आर गिविंग ना, बिकॉज़ पेपर्स आपको या तो सिर्फ एमई पे मिलेंगे या जीजी पे,
जी
बट आई एम जस्ट टॉकिंग अबाउट इफ ही इज प्लानिंग फॉर हिज थीसिस वर्क और समथिंग लाइक दैट ना, तो दैट कैन बी डन। आई थिंक दैट कैन बी डन।
जी।
Okay.
Yeah, thank you.
Thank you, Dr. Preeti and Dr. Subhash sir. Now further enhance
you,
this
sir.
section with the practical aspects of how to install R and basics of R. So now the lecture will cover by Dr. Kapil Choudhary. So Dr. Kapil Choudhary, you can start your presentation. Thank you, Subhash sir, for joining us.
ओके, ठीक है भाई, थैंक यू।
Okay. So my screen is visible. Is it?
Visible.
Okay. Okay. So basically, in today's class, we will only discuss about how to install R and what are the basic commands and what are different package and function, all these things. From tomorrow onwards, you will study about the design of experiment, correlation, regression, and all these analysis like you discussed now. So today we will discuss how we go for, first of all, install the R. To install R, we have to install two things. First R and another is R Studio. You can also work in R also, but it is in advanced version of R, that is R Studio. So to install R, first of all, you have to go on crane.r project. His is there. I think so. One second here. And according to your Windows, like if you using Mac, Linux, Windows, suppose I am using Windows, we will go for the window. Next, we'll install R for the first time. Then it will ask, means for Windows 64-bit and all this. So these are the basic steps which we already mentioned in this PDF file. So first of all, you have to go through this and you have to install in your system. Okay. So in all these first page, you have seen that first of all, we have to install only R. Okay. Once you install the R in your system, then we will go for the R Studio. Like up to this, I think so. Yes. So you will install only R. After that, you go for the R Studio. One sec. Hello.
Hello, sir.
Yeah.
So once you install the R, then you will go for the R Studio. Okay, this is the R Studio. Okay, so what is the benefit in R Studio over the R? Why we go for the R Studio instead of R? Because suppose I install the R and my interface is like this one. Okay, will be my interface. So it is R Studio. Okay, so there are 4 windows. It is first window, second, third, and fourth. Okay. These are 2 windows where we mainly go for the programming and coding. So this window we call as a editor window. Here, like it is like a text editor where we can I can write my name. I can edit it also. It's like as a text we will work here. But in the below there is another window is that we call as a console window. So whatever we perform in this window, in editor window, exactly will appear here. If you go for only R, then you have to write here, like if suppose I want to means multiply 2 into 5. Okay. Now if I want to edit it, so again I suppose again I multiplied 3 into 5. 3 into 5. Okay. But suppose I want to edit 2 into 5. So I have to go upside like this one. And again I have to edit it. But suppose here I written 2 into 5. 2 into 5. 2 into 5. Suppose I want to edit here. Now only on a single click I can edit my any command instead in like back and forward. Okay. So that's why we mainly use R Studio instead of R. Okay. First thing is in my right side there is another two windows there. First is environment window and another is like window which there are many functions like file, plot, package, all. In environment window, what is appearing is whatever work we are doing, that will appear in here on in this session of environment session. In history part, they will show you how you means recently if I maybe run a heat map and like if I multiply 2 into 5, whatever the history you have appeared in your editor version, or maybe it may be in console portion, that will be appearing history portion of. So basically there are two portions, that is environment and history. Suppose I want to import some data is there. We will discuss how we will import and all this. So there is another option is like import option is there. Suppose my data is in text format. Suppose my data is in Excel format. In any format, we can import from here. Something appear, if I want to clear it now, we can swap it, clear all the object from the work office. Okay. So this is my environment window. In below the environment, there is different menu like files, plot, package, help, viewer, presentation. Okay. So whatever, suppose I plot something, that will be appear here. Okay. So after once my plot is appear, you can save it as an image, you can save as a PDF, clipboard, everything. Okay. Now there are another menu is package. In package, what we do? There are, I think, nearly 19,000 packages are already in current software. So you have to first of all, if you want to work any package, suppose I want to work on heat map like this one. If I want to go for correlation, if I want to run a linear model, so for every function there is one package is there. Okay. So first of all, you have to install this package. So how you will install this package? There is one option is there, like this is my one option. Okay. Now suppose I want to install like read csv.bulk. Any I can install any package with their all the dependencies. Okay. So this is the how we install this package. It will take 1 or 2 seconds maybe. Package is big, then maybe 10 to. So these package, once it is installed, now I can search here. Read. Okay. So there is my package here. Now, if my package already installed, but you have to import that package in your R Studio. So for that, you have to click it. Okay. Then it directly will be imported in your R Studio here. Okay. And whatever we work do here means if I multiply 12 into 5, everything will appear in console only. Okay. So but we prefer R Studio. That's why because it here editing is easy than the existing R. Okay. So these are the basic 4 window where we perform many tasks. Okay. In first window, editor window is there. There are 2 or 3 options there. Suppose if I want to run a command line, any command, whatever line we write here is as known as a command line. Okay. Now suppose you want to run it. There are 2 options for the run a command line. One option is either you can go to and run and click it. So it will be directly done. First option is there. Second option, you have to put your cursor on in between or maybe last first in this command. Okay. And after you have to click Control plus Enter. Control plus. That again also appear here also. So these are the basic to how to we run in command in R Studio. Okay. So this is my editor version, editor windows. Okay. In editor window, suppose I'm working here. Suppose I'm working here only principal component analysis only in this window. Suppose I want to work in different data or maybe correlation and everything, but I want to make a different file. So to make a different file, you have to go here. Here this one window will be appear that R script. Okay. It will be added another R script. here you can work on correlation, regression maybe. Okay, so in this way you can proceed further to add a new script. Okay. In file option, there are, if suppose I open a Excel file or Word file, where we see there's a new file, open file, close file, save it. All these menu are there, but they will add on some extra menu because it is R. So that's why they add as a new project and everything. Okay. So these are in mainly which thing is useful for you, that is view. Suppose I want to, it is my normal view. Suppose if I want to zoom it, okay, then it zoom out. Okay, suppose I want to view in actual size, this is my actual size for this one. Okay, then plot section is there. Suppose you want to zoom your plot, zoom out your plot, and all this thing. Okay. So these are the basic interface which we, whenever you install your R, then we will face it. Okay. Now in R, first of all, what is the R? As I discussed earlier, that what is R and why the name of R? Because they are given by two scientists, Ross and Robert, at University of Auckland in New Zealand. So their initiation has taken as a R software. Now it is in a open software, means as I shown in PDF, you directly go and install your software, R Studio, and work there. So it is mainly open. If I am expertise in R, so I can upload my package. These are the packages which we have uploaded. Suppose I am working on forecasting. Suppose my work is forecasting. So here this one. So these packages, different packages are there. So we develop all these packages. Okay. So in like EMDTD and all this. So we have to, I can upload any package if I am an expert on that subject, and if I develop any model, and it is also for a community, a research community, good for the research community. So it is a basic. What is R? Now, what is the limitation of R? R mainly is case sensitive. Means suppose if I write X equal to, it is my capital X, sorry, capital A. Capital A equal to 2. Okay. Then A equal to 2. Now suppose I want to print small a. Then in my console, the error will appear that object A is not found. Why? Because R is a case sensitive. So whenever you're using any text is there, so you have to be conscious about the case sensitiveness of the R. So if I print A, then directly it will be printed. Okay. So it is the basic R. Okay. Now, if how we go for the command, what is the command? Command is nothing but the instruction which we give to computer or perform to any set of instruction which we desire. Okay. So this is the command. So first line is my command. Okay. Second line is my command. So all these are command. How we execute all this command? So it is mainly executed. If I write in the console, then we directly get, like if I write in console 2 into 5. Okay. 2 into 5. So directly to get it. But in how we write in R, directly in R, you have to write directly, nothing required. Like if I multiply 2 into 5, run it exactly. So in command line, you suppose if I want to jump over the next line, there are two ways. Either I can use as a semicolon, semicolon, and go for the next line, or otherwise I have to switch on the next line by using int. Okay. So these are the command. You know, and in command, actually every line, suppose if I given 2 into 5 in name, I want to give a name. Okay. Suppose I give a name A, then it will A equal to 2 into 5. Okay. So first of all, I have to run it A into 5. After that, I want to say what is the A, value of A. So I will only enter A and press run, or either it may be control plus enter. Yes, A. So it appear in console, that is out. Okay. So it is the basic output, and is how we perform in our all this. Now, if like in Word or Excel, we set a directory where we want to work. So suppose I am working here. So what is my working directory? Okay. My working directory. So my working array get my working ID, working directory is this. Means I think it is visible to all of C user Kapil desktop GPV. Okay. In that I am making a folder, and there I am working. If you want to save all these things elsewhere, then you go for the set working directory and proceed further. Means here you have to give your location. Suppose I want to say on C users, backspace, C users, like Kapil, and only desktop. Okay, desktop, or you can write anything, only your folder's address you have to insert. But we remember that R use as well always as a backslash, not as a forward slash, because whenever we go for and copy anything, like suppose in GPB and I go for this package, and if I am going to search the address of it, so one second. Yes, this is my address of the mesh file. Okay, C/user Kapil desktop GPP. Okay, these are the address of my this file. But in R, we always use backslash, not front cell forward slash here. Okay, so we cannot use directly address of that particular location, but you have to change or modify it, and after we have to proceed. So there you can go and set your working directory also. So this is the basic working directory. Now, what are different type of data we use in R? First of all, how we use different data. So mean different data says we use mainly vector or a data frame or a matrix data or maybe your Excel data, CSV file, all these files we use here. But for the initiation, suppose I have a vector. Vector means it will consist only some numerical value or maybe some character also consist, it may be consist. Suppose I define my vector as a X. Okay. To start anything like this, if I write as a C and in bracket, so it will be my function. It will be my function. Within the function, I will write something. I will give some instruction to it. Okay. So for every package, like suppose here we go for like this is a package. Okay. So one second. Like this is package. So it is a package EMD, ELM. Okay, this is the package. But here are different functions. Like first function is EMD, ELM. Okay, below it. But maybe as EMD, EMD, ELM, all this. So these are the different functions. So first of all, you have to understand difference between the package and function. The function is small thing and package consists of all these functions, particular on the particular themes. Okay, suppose I make a package on ensemble empirical mode decomposition and its variant based ELM model. Suppose it is a forecasting model. So I develop, there are three functions. Okay, means three model I developed, but my theme is same. So on the name of this package, I uploaded the name of package. Okay, so here first of all you have to install the package, and inside the package there are different functions, and according to your choice you have to use any function out of this. Okay, so this is the vector. To how do we form a vector? Like suppose I want to enter like vector 12, 23, 25, 36, all these things. Okay, 67. So these are the my vector. Okay, so if I run it is once your any command run, then it will be in blue line. Okay, suppose it is error, something is error there, then it will be appearing red line like here, error in C. Why? Because I left that comma. Okay, so it is basically C is the function. It is the basic function and we have to perform it. After that, C is the basic, but suppose I want to insert a matrix data here. Okay, so in matrix, to run any function, you have to go and search anything, and after you have to put your cursor on that, and that will be give you instruction like matrix data, comma, number of row, number of column, by row, all these things. Okay, so suppose in matrix I insert as a data like this one, I insert as a vector. Okay, this is will be my vector. Okay, and I suppose that number of row equal to 2 to 4 5 56. Okay, number of row maybe 3. Okay. Now x equal to matrix. Now I want to see what will be my x. So suppose I want to run it x. So it will be taken as an entry as a in 3 by 2 matrix. Okay, 12, 23, 35, 56, 67. Means it taken as a data is serial wise. It is come under the first column, it comes under the second column. You also can change here. Suppose I want to add number of column, okay, number of column equal to 3. Okay, so and again I want now, instead of there are 2 columns, there are 3 columns. Okay, so as you give your instruction, and you can edit anything. Suppose I want to edit here my first thing, then you can go and put your cursor and edit it. But you cannot, suppose if I want to edit in this line, suppose this is my line, how I will edit here? So for this, you have to put your cursor here, put up arrow like this one, and you have to reach this. So it is time taking thing. So that's why we prefer RStudio instead of directly R console. Okay, so these are the matrix and vector form. Now suppose I want to add some data like my age. Suppose I have taken a data of 10 student age and weight. So suppose I have taken data as age equal to, first of all we have to create a vector age maybe 20, 23, 25, 37, maybe this. Okay, so it is my age. Now suppose I have taken their weight also, corresponding weight also, then weight could, I can name anything. You can name here anything. Okay, and suppose if you want to whenever we write any code, we want put some instruction there. So here, if you want any instruction, put this thing. Okay, and after, like if I want to, if we want to form a data frame, so everything is in green, means it is not a command line. Okay, it is basically text line that will be not run. If suppose I will run here, then it will directly jump on the next line. This line will not be run through a simple form, like if you use a run or control center. So if after age, if I will add a weight option, suppose I go for weight for 20, 45, 67, 58, 54, okay, 44, 5. Okay, these are the 8 my age and weight. So first of all, you have to run your age. After, you have to run your weight. Okay, now I want to make a data frame. Data frame is nothing but is a 2-dimensional frame where we put age and weight side by side. Okay, so suppose my data frame equal to, I say my name, given as name is data. After that, you have to run a function. First of all, you have to run a, there are different functions, out of which I want to select only data.frame. Okay, data.frame is open. Now under what things will become? Under age, comma, your weight. Okay, weight. Once I run it, okay, now if I want to see what will be my data frame, so I have to run it. Either I have to write here data, because I named this as a data. Okay, otherwise you also can go and only select the data portion, like this portion. Okay, this I want to run it. Okay, go and run option. Complete. Here you will be data form, means data frame, whatever we formulated here for your age and weight for 5 students, we put here. Okay, now suppose I want to see its head. Suppose it is my small data, 5 data, but in your genetics and plant breeding, you use thousands of data in Excel file. So how you will see whether you exactly correct data or not? So for it, you want to see its head and tail. Okay, suppose I want to head of my data. So my data name is data only. So I have to give a name of data. Okay, and run as a head. So it will take directly exactly 5 data. Upper head portion of, suppose I have to add here more data. Okay, like this one. Okay, again we have to run it. Means once you edit anything, first of all you have to run. After that you have to proceed it. Okay, so again, so I have to run it as weight, data, and my head data. So it appear only upper 6 portion, upper 6 only. Okay, but my initial data was there are, suppose my 12 point are there. Okay, there are 12 points, but out of 12 they've taken as a head as 6 point. 1, 2, 3, 4, 5, 6. So 1, 2, 3, 4, 5, 6. Now, if I want to say see my tail of the data, tail of the data, given data set. So these are the below 6 point, okay, 7 to 12 point, okay, whether your data is correct or not. So all things you have to see first. Once you import a data set from either it may be CSV file or Excel file or text file. Once we will see here how to we import all these data, okay. So now how we import all these data? There are different method. Means either one method is to directly you have to write the code. Another way are means their simple and foremost means very simple method is first of all you have to go to import environment section. There you have to go to Excel file. Okay, suppose my file is Excel. Okay, now I have to go there and any folder you can search, like in D folder you can search, E folder, any folder you can search. But suppose I am on GPP folder, okay, and my data is GPP. GPP dot suppose maze.x, okay. So here one option is there given is import, means you have to import it. It is univariate data, as Subhash had told that there are different data like univariate, multivariate data is there. So I have taken as a univariate data. We will also take as a multivariate data also we will take, okay. So I have taken as a data is maze, okay, imported data. So now you give a name to that imported data. Suppose I imported a maze. So maze, or yeah, you can give my data equal to what is your name of imported file. Here they have written as a imported file name like maze, okay. So once you run it, the line is command line is green, sorry, blue. So means you are exactly correctly imported your data. So it is the simplest method to import anything. Suppose I want to import a text file. So go for a text file. Suppose you may use a SPSS, SAYS, and data file is there. Means something have saved in the form of output, but you want import here and perform some different analysis. Maybe it's SPSS or SAYS. So you also can perform it. Okay. So whatever thing we have means analysis here, that exactly appearance in my environment section, okay. So now suppose I want to calculate some basic statistics about my data. Suppose my data is maze only. Now I want to calculate mean, median, mode, all these things, okay. Suppose I want to calculate mean of my data. Mean of data name is my data, and this, sorry, mean, one second. First of all you have to run it, and you have to run a summary. In summary, everything will be there. My data. This thing, okay. In summary, everything will be appeared like minimum value, first quartile, a median, mean, third quartile, all these things. Okay. Suppose I want to plot my data. Okay. So for the plot, you have to go and write here. Means in the bracket, you have to give instruction. Whatever they are asking, like in plot, suppose I have plot, they are asking what is the value of X, Y, all these things. So either you can also search here. Like suppose I want to go for the summary. For go and help file, search here like summary. Okay, summary. This is my summary. So they will search all these result regarding the summary. Suppose my file is something there. ANOVA is there. So ANOVA is there. So here they use a summary. Okay. So in that summary, what is the written here? It is your function. It is your function. ANOVA is your function. And in curly bracket, they have written car. Car is your package. So to run the ANOVA function, first of all, you have to run the car package. Means first of all, you have to install the car package. Once you install the car package, suppose I install the car package. Maybe there. Car is there. Okay. It is my package. Okay. I will run it. Okay. Now I will search here car. There are two options to bring this car package into your working directories. Two options. Either you can go for the library function, like library, and write down here your package name car. Okay. Once you run it, your direction of package will be directly tick marked. Okay, means it has been directly imported in your working direction. Okay. So here we will working as a plot for the my data. Okay, so for the plot, you have to go for here and type your data, my data. Okay, it is actually, I have not taken it, converted in time series data. First of all, you have to convert in time series data, and after that, you have to proceed with it. Okay, suppose so this one. So first of all, you have to convert your data into time series data, and after that, it will be directly read as a, and here it will be plotted. Okay. Once it is plotted, now you want to export it as, like if I want to export as a image as a PDF as a clipboard. Suppose I want to as a copy clipboard option. Okay. In clipboard option, they will give, you can also change your size, height, weight, everything. So here directly you have to drag it. Like I want this one size. Okay. Suppose small size. Go for it. Okay. Once it is okay for you, then go and here you make right and click copy your, and now you can paste anywhere that function. Suppose I want to paste in my Word file. Okay. Once again. Once. So you can save anywhere, okay, where you want. Now suppose if I want to import as an image, okay, so save as, but it will ask your location. Okay. Here you have to give the, it is my file name. Suppose I want to change it as a directory and I want to save as a desktop. Okay. Desktop, open, okay. R.C. It is in JPG format. Here your already plot is too. Okay. So there are different way to save any plot here. So actually today is the basic class, how you mainly import the data, how you perform the analysis, everything. Analysis wise, we will proceed tomorrow onwards. So it is the basic note. Once all these things done, now suppose I have, it is my Excel file and I have done all these analysis, but suppose I have CSV file, how I will run it? There are different, either you can go from here, another way is, here you can see, suppose I give my file name is a CSV file equal to read.csv. There are different way of read.csv. Okay. There I have to write it file.choose, okay, and run it. Now they will ask where is your file CSV file. Suppose it is my zip file is there and it is there. So I have to open it and already file has been done. Okay. So means my file has been imported from as a CSV file imported in my studio. Once it imported, now if I want to see what are the different data in CSV file, I have to exactly select the name of the file. Okay, I given as a name as a CSV file. Okay, and I have to run it. Once I run it, all data appear here. There are different 4 biological data are there, like PP means plant population, plant height, number of green leaves, and yield of different. How many? There are 46 sample plot are there, and among them they have collected 4 different variants. Okay. Now suppose I have selected all these things. Now I want to work only this particular PP, means plant population. I work only if I want to work only plant population. Then how we proceed here? To extract out of 4 variable, if you want to extract only plant population, suppose I give my magic plant population equal to, first of all you have to give your existing file, like my existing file name is CSV file. Then you have to break it, big bracket, and out of 4 variable, if I want to extract only first variable, then give the column number. It is my first column. Okay, so has been extracted, like pp equal to CSV file, it has been extracted. Now suppose I want to extract, next is pH. Okay, then nothing you have to, just you have to directly copy paste here and write here another name pH. Okay, in CSV file, but it is my second column, so it will be my second column. Okay, run it, then already run. Okay, third is your NGL and yield. Okay, so that will be your NGL, number of green leaves. Okay, it is also, we have to extract from the CSV file, but it will be my third column. Okay, so NGL file. Now finally I have to extract yield. Okay, again you have to give your file name. It is my file name, and it is my fourth column. Okay. So all these data I have extracted. Now suppose I want to perform, actually all these will detail discuss in next classes. So today we only discuss about the basic everything. So suppose I want to find out the correlation between the yield and plant population. Okay, so you have, for this you have to run as a correlation. Okay, correlation between your PP comma yield. Okay, this is the 0.385. Okay, so suppose I want to change that, I want to find out the correlation between pH and yield. So what will this one? This is my pH and yield. Okay, and if I want to find out the whole correlation is among all these 4 variables, so for that you have to write here on the correlation and your file name CSV file. Okay, so out in output they will show you that PP, among the PP and PP they are exactly one correlation, but among PP and PS 0.239, among PP and NGL there are 0.28, and yield and PP 3.38. Okay, so they are the different correlation. Now if I want to plot is all these 4 variables, for that I have to use as a function pairs, and again I have to write my file name CSV file. Okay, so in the plot function they will show you among correlation between different 4 variables. Okay, so for that means, first of all you have to search which function we will use for this. So either you can go and help from Google. Suppose I want to find out the correlation between the 2 variables. So exactly, if you don't know, if you know, then this is good thing. Find out the correlation between two variable in R. So there definitely they will show you functions names. Okay, suppose I here is there. There are different functions like this one, like core computes the correlation coefficient. Okay, so you have to use this function. Now how you will first of all you have to install the package which consists of core function, and after that you have to perform. Okay, so it is a basic how we code. Suppose I want to find out the regression. Okay, for the regression what we do, like for the regression you have to find out, or you have to run as a function as a linear model. Okay, in linear model you have 2 variables. One is our independent variable, and another is dependent variable. Okay, so here my dependent variable is yield, like my dependent variable is yield. Okay, and you have to give this symbol. Okay, it is my independent. Now my dependent variable is, suppose my dependent variable is only one variable. It is my simple regression model. In simple regression model, only one independent variable is there. Okay, and if I want to run it, already run. Now, if I want to see what is the output under the rig. So in the model, intercept coefficient is 0.83 and the PP coefficient is this one. Now suppose I want to run instead of a simple linear model, if I want to run a multiple regression model. For that, you have to add some more independent variable. Here my different 4 independent variable are PP, pH, and plus NGL. Okay, so these are my 3 independent variables. I run it. Now, if I want to see what are the output in regression, it is my output means basically they are giving different coefficient of each variables. Okay, if they are using intercept, means for that you have to use minus 0.0840 and for PP 0.102, for pH 1.6. All these are the coefficient. Suppose if I want to see the summary of this model, summary of my linear model, sorry, summary of regression. My variable is regression. Okay, so it is the output, means they have used different coefficient, their standard value, their t value, their p value, and instead of each variable they have mentioned one star. Like in star means it at what level of it is significant, like 0.1 means 10%, 0.5 5%, 1%, 0.1%. All these are the significance level. Out of these 3 variables, you can state that 2 variables are significant at 10% level of significance according to your data. Okay, there are residuals, standard error are this one. These are 42 degree of freedom is there, and multiple R square. Mainly R square means explained variation, and due to these independent variable. Okay. So these are the basic how we run all these things. Now, last thing come in our mind that how we will, once you have imported your data, okay, you have done some analysis. Now, whatever the analysis you have done, you want to export it also. Means you want to save here somewhere else. Okay. So to save here, there are the 2 things. One is, suppose I want to save yield only. Okay. This maybe my output is there. Okay. These are the data and which are my output. Now, if I want to save or as a export it as somewhere else in my local directory. Okay. So for that, you have to write .csv. Suppose I want to save in a CSV format. Okay. Write .csv. Give the file name. Okay. Means what thing do you want to means from here do you want to export it? Suppose I want to export yield. Okay. So write here yield. Okay. Yield. Okay. Comma. Give your file name. File means at what name do you want to save it? Suppose I want to save name as a my name. Okay. Save and run it. Now file has been done. Now where it is stored, I think my working directory is this one. So here the file has been stored. One second. So here it is my file and file for the export. Okay. You also can save in an Excel file or Word file, anything you can save. Okay. So now another option, suppose whatever analysis I have done, I want to save all these things. Okay. So for that, you have to run here as a sync function. Sync, and you have to name the, sorry. You have to write here sync, and in the bracket you have to name any file. Like if I want to mention a name of like residual or result file, result.doc, and you have to use double semicolon. Okay, and you have to run it. Okay. Okay. After that, you have to run as a sync, okay, and run it also. Now, whatever things you want to do analysis, exactly you have to run it. Like suppose if I want to run first X, then it is this age, weight, or everything. Like age, weight, data frame, height. Okay, so everything will be stored in the name of one result.doc file. One second. First of all, you have to open it. One second. So first of all, you have to write this sync file, and after you have to do any analysis. Suppose if I want to calculate the mean of this one, okay, mean of age file, mean of this file. So first of all, go there, search whether my file has been generated or not. It is generated. Okay, file has been generated. Now I have to calculate the, suppose I want to calculate the mean of age. Mean of age. I think maybe problem in that, but we'll see in next class. Okay, this function. So it is a basic how you import your data, how you perform different analysis. Part of analysis will proceed in next classes, but it's the basic. Okay. So to save your time, it is already done. Suppose I have some another data, and if you want to perform something else, cluster analysis and all. So basically, is story of how do you run a function. Okay? If you have any query in the basic of R, then you can ask. Otherwise,
So if you have any doubt, you can ask. Yeah, David Thakur.
हेलो सर।
हाँ हेलो
नमस्ते सर।
नमस्कार।
सर हमें ये कैसे पता चलेगा सर कि कौन सा फंक्शन किसके लिए यूज करना है, जैसे कार पैकेज सर, जैसे कार पैकेज आपने नोवा के लिए, कोरिलेशन के लिए, इसकी लिस्ट सर कैसे, कहाँ से ये पता चलेगा सर?
देखो, ये अगर आपको कुछ भी नहीं पता चलता, जस्ट हम लोग क्या करते हैं कि हम लोग ये तो सर्च कर लेते हैं कि हमारे पास फंक्शन कौन सा, यही पहले पता करना है आपको, ठीक है। तो इसके लिए दो ऑप्शन है। एक तो ये है कि जब भी हम आर स्टूडियो ओपन करोगे आप लोग, उसके
यस
हेल्प
सर।
फाइल में जाके सर्च कर सकते हो। पहला ऑप्शन, लाइक आपको अनोवा ही सर्च करना है मान लो, ठीक है,
यस
कि
सर।
मुझे अनोवा रन करना है, मुझे रिग्रेशन रन करना है, मुझे ये रन करना है, तो हेल्प फाइल में उसका कीवर्ड आप डालिए, ठीक है।
ओके सर।
उस फाइल का जो भी एग्जिस्टिंग, लाइक अनोवा कार में है, अनोवा कहीं और पे है, तो जो एग्जिस्टिंग फंक्शन है वो आपको लिस्टेड हो जाएगा। पहली बात तो ये, अगर
Okay,
वहां
sir.
से भी नहीं पता चल रहा तो हम लोग जैसे सपोज करो हम गूगल से सर्च करते हैं कि इस फंक्शन ये मुझे एनालिसिस करना है, इसके लिए मुझे कौन सा फंक्शन यूज करना है, तो वो
Yes.
वहां मेंशन करेगा कि ये पैकेज आपको यूज करना है, ठीक है ना। और जब भी आप किसी पैकेज को यूज करने जाओ,
Yes,
जब भी
sir.
अननोन पैकेज है मान लो आपके लिए, तो सबसे बेस्ट तरीका उसमें, जैसे मैं आपको शेयर करता हूँ सेकंड, एक बार रुको, डाउन से सेकंड, एक सेकंड मैं आपको बताता हूँ कि एक बार मैं फाइल, हाँ लाइक वन सेकंड। Okay. So here you can go any for, suppose I want to run this function like a bind or any, like if I want to agricole, you mainly use agricole function for agriculture strategy. So in agricole there is a different function like me, like we are discussing now.
Okay, sir.
So you go and here search, suppose in design, you go and search here like RBD design or CRD design. Okay. In CRD design, they have uses. How do you use it? Like first of all, you have to write down this one. After you have to mention your treatments. How many treatments are there? How
Okay.
many replications are there? How many seed means how many randomization is there? Okay. And what are the method of randomization? Whether randomization done or not? Okay. In the last part, they have given example, basic example. Okay. You have to run it also. Okay. Means it's a basic thing. Before run any new thing your part, first of all, you have to run, if you are new to this region, so first of all, you have to run example. If you will understand through the example, then it will be easy for you so that you can perform different analysis also. Okay. So there are two ways. Either you go for help file and here you can search like CRD. Where is my CRD? Okay. They will
Okay.
show different CRD option. They will show you like there, like design, agricole. Like I search only CRD. Okay. They mention agricole double seven design dot CRD. My first name is agricole is my package and next name is my function. Okay. Like this one you can search.
Thank you, sir.
Okay. Okay.
Anyone
If you-
else?
सर
हाँ बोल।
सर प्लीज ईमेल से हम लोग को ये एक्सेल फाइल भेज दिया करें तो हम भी अपने सिस्टम पे प्रैक्टिस कर लेंगे सर, ऐसे तो समझ में नहीं आ
हाँ
रहा
हाँ जरूर जरूर जरूर, हम आपको स्क्रिप्ट भी भेज रहा हूँ अभी, जैसे जो भी आज परफॉर्म कराया उसके, और साथ में मैं डेटा भी भेज रहा हूँ आपको, ठीक है ना।
है। हाँ सर, स्टेप वाइज सर करते जाएंगे तो सीख जाएंगे, अदरवाइज
हाँ जरूर
निकल रहा है
जरूर
ऊपर। थैंक यू
जरूर
सर।
जरूर।
So please provide us the list of basic packages in R which we can use for genetics so that we can install them before the class. It will be useful for us.
Yeah. But actually, in further classes, we will show you which package we should use for which analysis, like genetic
Okay.
correlation, PCA, stability, everything. Means a little bit we will cover in our next lectures. In this class, we just showed means the basics of R. Means this is like
Okay, sir.
open source software, so we are covering mainly R software. There are other many tools, but we will cover mostly those are free. So thank you,
Okay, sir.
Dr. Kapil Choudhary, for your wonderful lecture. And this session is over now, and we will meet tomorrow at 11 a.m. Thank you, everyone. Thank you so much.
Thank you, sir.
Thank you, sir.